Right, the last section of the notes for this chapter on microbial metabolism deals mainly with fermentation. And so your objectives for this section include comparing and contrasting aerobic respiration, anaerobic respiration, and fermentation. So each of these three. Looking at some of the uses of fermentation, and then finally just kind of finishing up with what are some of the different nutritional patterns of organisms. How do they obtain their nutrition? So some different vocabulary that we'll have you go through. So if we look at respiration again, first of all, we always start with glycolysis, where we break down glucose, we release some NADH, and produce a little bit of ATP, breaking that glucose down into pyruvic acid. Now, if there's oxygen present, then we go into aerobic respiration, and then that intermediary step where we have some CO2 released in NADH to produce that acetyl-CoA, that 2-carbon molecule that will enter the Krebs cycle to produce some more carbon dioxide, some more ATP, and some more electron carriers. Those electron carriers from glycolysis and from the Krebs cycle go to this electron transport chain, and that's where we get most of our ATP being produced. We also have oxygen as our terminal electron acceptor that then is combined with those hydrogen ions to form water, which is another one of our end products. Okay? But if there is no oxygen present, then organisms are going to use fermentation. And they're going to take that pyruvic acid and they're going to break it down into some different types of fermentation end products. Now, this can happen in our own cells through lactic acid fermentation, but bacteria and yeast, they can carry on fermentation and do what's called alcoholic fermentation to produce alcohol as an end product. So we'll take a look at that. So when we look at fermentation, some of the definitions are spoilage of food by microorganisms. Okay, those things have fermented. Processes that produce alcoholic beverages or acidic dairy products. Again, we use fermentation to do that. And then large-scale microbial processes occurring without air. Okay, and so when we look at the scientific definition, we're looking at the oxidation, release of energy from the oxidation of organic molecules. Does not require oxygen. Okay, so if there's oxygen available, pretty much all organisms, including bacteria, are going to use aerobic respiration because it enables them to make a lot of ATP from just that one glucose molecule. However, if there's no oxygen available, then they can use fermentation. So then we don't have the Krebs cycle, we don't have the electron transport chain. In this case, there's an organic molecule that acts as the final electron acceptor instead of oxygen. So alcoholic fermentation is one of those types of fermentation and occurs in bacteria and yeast. And what happens, we always have glycolysis first, okay, to break down the glucose into pyruvic acid, but then that gets converted to ethanol and carbon dioxide. And so if it's a heterolactic type of fermentation, it's going to produce lactic acid and other acids or alcohols. Okay, so this is important in the baking and the brewing industries. So the CO2 end product is important for the baking industry, and the ethanol end product is important in the brewing industry. Lactic acid fermentation can occur in your muscle cells. So if you've worked out and really run a long race or whatever, and later on your muscles are really sore, well, it's because some of that lactic acid has built up. Because if you're working out really hard for a long time, you think of a marathon runner, they've been running and running and running for those 26.2 miles, or somebody who's been doing a really fast sprint and they've run as hard as they can, they've used up so much oxygen that sometimes they can't get oxygen fast enough to their cells to complete the aerobic cellular respiration. And so if that oxygen isn't getting there quick enough, then our cells may switch to lactic acid fermentation. Well, in that case, we start producing lactic acid as an end product. And if that builds up in your muscle cells, it causes that muscle soreness. Now, if bacteria cells carry it on, they can actually use that lactic acid end product to help in the souring of milk, to produce yogurt. They can produce sauerkraut from cabbage, pickles from cucumbers, kimchi, I think is also fermented. I don't know if that's cabbage with something else. I'm trying to remember what kimchi is, but so there's a lot of different things that we can use lactic acid fermentation to help produce. You know, you think of the tartness of sour cream and some of those types of things, and the tartness of yogurts, all with that lactic acid fermentation. Certain types of cheeses. So it converts that pyruvic acid instead of to water and carbon dioxide like we do, and ATP like we do in aerobic respiration. In fermentation, it produces lactic acid. And so the two main types of bacteria here are the Streptococcus species and the Lactobacillus species. So if you even look on the side of a yogurt container, you're going to see maybe some Lactobacillus species names on there, and other types of bacteria listed on the yogurt. If it's a homolactic type of bacteria, it only produces lactic acid. So here you can see we still get 2 NADH, 2 electron carriers produced, and we get 2 ATP produced from glycolysis. So that doesn't change when we break the glucose down into 2 pyruvic acid, excuse me. But once we break that pyruvic acid down, we need to use those 2 NADH here to further break that down into the end products. So these get recycled back to glycolysis. So fermentation basically allows glycolysis to keep continuing because it keeps providing these electron acceptors back up here for glycolysis to occur. So fermentation itself does not produce any ATP. Glycolysis does, but fermentation does not. No ATP. So it is in the best interest of the bacterium to use aerobic respiration if it can, when oxygen is available. So again, some of the things that can be produced from some of the different bacteria: lactic acid, ethanol and carbon dioxide, propionic acid, acetic acid, butyric acid, butanol, acetone, isopropyl alcohol that we use in our rubbing alcohol when we spray down our desks, succinic acid, acetic acid, formic acid. So all sorts of different types of acids produced depending on what type of bacterium that we're working with here for those fermentation end products. Okay, so again, lactic acid fermentation occurs in some bacteria and within your muscle cells. Alcoholic fermentation occurring in bacteria and yeast cells to produce alcohol end products. Now, one of the tests that we will do, and we're doing this in lab on Friday, is what we call our fermentation tests. What we use is we don't use these Durham tubes, but we do use a pH indicator that turns color if the acid is produced. So we use a phenol red indicator. So all the tubes start out red to begin with, and you're going to inoculate your bacterium in each of the tubes. And we use phenol red sucrose, phenol red lactose, and phenol red dextrose, which is glucose. So you're going to have a PRS tube, a PRL tube, and a PRD tube. They'll all look red to begin with. We're going to have you put your bacterium in there. We're going to let them sit over the weekend. But if they turn yellow, any shade of yellow, that means that lactic acid is produced that changes that phenol red to that yellowish or orangish yellow color. Okay? And if we would have inserted these little tubes here, these Durham tubes in here, if any gas was caught in there, so that air was trapped in there, that means that they release CO2 as well. So they produce lactic acid and carbon dioxide. So this helps you identify and differentiate bacteria. So it's going to be one of our tests we use, because you'll see that different classmates who have different bacteria, their tubes will turn a different color. Some of them will stay red, some of them will turn yellow. Maybe all of yours will stay red, maybe all of yours will turn yellow. And so it's a way we can identify the different types of bacteria. And then we also have a little bit on lipid metabolism and what process they go through to get broken down. The different proteins, carbohydrates, and lipids as far as their breakdown. We pretty much focus on the carbohydrates when we're talking about metabolism. And so lipases, again, those are enzymes that break down fats into fatty acids and glycerol. And then proteins, they get broken down by enzymes called proteases and peptidases. And then there's some terms that go along with that. Deamination, that's when the amino group of the amino acid is removed. That's the NH2 amino group, and converted to an ammonium ion. Decarboxylation, removal of a carboxyl group. That's that COOH group. And then dehydrogenation, removal of a hydrogen atom. So some terms that go along with protein catabolism. So again, a little bit of a summary here. Cells have to have an electron donor that serves as the initial energy source. So, glucose, if we're going to break down glucose, it's going to donate some electrons to those electron carriers. The electrons removed from the chemical energy sources are transferred to electron carriers. Some energy gets produced. Electron carriers are transferred from electron carriers to their final electron acceptors, producing more ATP. Remember, in aerobic respiration, oxygen is the final electron acceptor. In anaerobic respiration, inorganic substances are the final electron acceptor. And for fermentation, organic compounds are the final electron acceptor. So this is where you get the most ATP. So most organisms will carry on aerobic respiration if they have the choice between all of these, if oxygen is present. The last little bit just deals with different ways of classifying organisms according to their nutrition. So if organisms get their energy source from a chemical, they're called chemotrophs. If they get it from light, they're phototrophs. So again, light is the energy source for a phototroph. Using breakdown of organic and inorganic compounds for chemotrophs, and then autotrophs. They usually use carbon dioxide as their carbon source. Heterotrophs feed on others, so they require an organic source of carbon for their energy source. Photoautotrophs use light as a source of energy and carbon dioxide as their source, so they're going to use photosynthesis. Okay. Chemoautotrophs use electrons from inorganic compounds as an energy source, and a carbon source is CO2. Chemoheterotrophs, that means these make their own food. Auto means making your own food. Heterotrophs means you feed on others. So they're going to get their electrons from hydrogen atoms in organic compounds. Saprophytes live on dead organic material. Parasites live in or on a host to get their energy. And then an amphibolic pathway, those are metabolic pathways that function both anabolism and catabolism. Photoheterotroph, that's a pretty big word. That means if they have light available, they can use it as a source of energy. But they use organic compounds, not CO2 as a carbon source. So they don't use carbon dioxide in the air. They actually feed on other organisms for their organic compounds. So lots of different terminology. Here are some of the different bacteria that fit into some of those, and other microbes that fit into some of those categories. So we use just some of those terms in terms of what bacteria get, how they get some of their types of nutrients to obtain their energy.